Hello, welcome to our video. In this video, we will talk about stem cells, the types of stem cells, and its application to the medical field. What makes stem cell really fascinating is that it has the ability to proliferate and differentiate into many different types of cells that made up a complex organism. A common misunderstanding of stem cells is that stem cells are cells that are programmed, bioengineered to survive in a lab. Well, it is not completely true because stem cells are present in everyday life. Your blood derived from a stem cells inside your bone marrow. A baby came from a stem cells, a cell that is made of a sperm and an egg, which then undergoes multiple divisions, which then end up to be making a full baby. Now, not all stem cells are alike.
Now most people think that the stem cell is a new discovery. It is only found to be in active debate only not so long from now, but they are wrong. Stem cell is actually found back then at 1901 by Alexander Maximov. Not in the picture, but he only found that the cell works as a certain precursor. Not much development done on that stage until the 60s. At that point, a new record of history had been made, where a leukemia patient were treated by using stem cell from her twin. It only works well for 6 months. The developments are followed with the embryonic stem cell isolation in 1981 from mice blastocysts. This development of embryonic stem cell is a very huge step in utilizing the stem cell, since a whole bigger concept of stem cell had just been opened on that point. All the development continues, and at 2006, the first induced pluripotent stem cell are found. It's found by Shinya Yamanaka from Kyoto, Japan, by using a mouse as the modeling, drug discovery and regenerative medicine, and many other scientific branches. The induced pluripotent stem cell will be main topic to discuss on this video.
In general, there are three types of stem cells, which are totipotent stem cells, multipotent stem cells, and pluripotent stem cells. Totipotent stem cells are stem cells that can form the embryo and the extra embryonic tissue. This extra embryonic tissue is critical to the survival of the embryo, which includes the chorionic villi, the amnion, definitive yolk sac, extra embryonic column, and many others. Multipotent stem cells are lineage specific and composed mostly of adult stem cells. There are two types of multipotent stem cells, which are the hematopoietic stem cells and the neural stem cells. Hematopoietic stem cells can give rise to the blood cells, ranging from red blood cells, white blood cells, platelets, and many others. And neural stem cells can form various types of neuron types, ranging from the relay neurons, motor neurons, and sensory neurons.
Pluripotent stem cells are the master cells which able to make cells from all three basic body layers. These three body layers are ectoderm, mesoderm, and endoderm. Ectoderm will give rise to the skin and nervous system. Endoderm will form the gastrointestinal and respiratory tracts, endocrine glands, liver, and pancreas, while mesoderm will form bone, cartilage, most of the circulatory system, muscles, connective tissue, and more. This type of stem cell has the ability of pluripotency, which has the potential to become any cell within the human body. Like all stem cells, pluripotent stem cells are also able to self-renew, meaning they can create more copies of themselves. Going deeper into the topic, the pluripotent stem cell can be obtained using two methods, the embryonic stem cell and also the induced pluripotent stem cell. The embryonic stem cell is the stem cell derived from the embryo. The cells will be obtained through fertilization and on its early development stage. The cell will be harvested on its early development on the blastocyst stage. The inner mass of the blastocyst will be filled with the embryonic stem cell, and the cell is what we require to get. The advantage of embryonic stem cell have high variation of medical possibilities, while the disadvantage of embryonic stem cell is that the cell is obtained by harvesting an organism's blastocyst, in which it will eventually die. Debates about this very issue has risen in many forums regarding about ethical concern of messing with life of organisms, especially humans.
Induced pluripotent stem cells. These stem cells is different from any other type of stem cells. How it is different? The answer is from the way to obtain it. Normally, the way to obtain a stem cells is by harvesting it on the development stage, which normally done to obtain embryonic stem cells. But in the induced pluripotent stem cells, a normal human mitotic cell can be from any organ or tissue will be modified using several transcription factors that will cause the revertation of the cell back into the stem cell stage. Take a look at the picture. The normal mitotic cell meeting the transcription factors of OCT4, KLF4, or the SOX2 will cause the cell to revert back to the stem cell stage. Considering the method to obtain the stem cell, these are the possible benefits from using the induced pluripotent stem cells. Firstly, similar to any stem cells, have high medical possibilities. Secondly, no ethical concern, since these cells is harvested from adult human who are willing to do so, and no harm is done to the corresponding human. Thirdly, very small chance of rejection in human users. If the stem cells is obtained from the same human, the rejection rate will be very low, since the cell match with the other cell found in the body of the receiver. On the other side, for the disadvantage, it is also present but similar with the other type of stem cells, where due to the very high possibilities that the stem cells possess on its pathway, unknown disease or side effect may arise from this usage.
Now we have learned about stem cells, the types of stem cells, and the function of stem cells. Now we will take a look at an example of stem cell application in skin regeneration. The skin may get damaged by burn, cut, or scratch, and it may cause a permanent damage that our body cannot repair. Through induced pluripotent stem cells, we can fix that. In the next segment, we will discuss the mechanism of those treatments.
So, in the human skin, it composed of several types of cells. For example, we have the fibroblasts, which found in our hair follicles. And secondly, we have the keratinocyte, which compose in the outer layer of our skin. Then lastly is the melanocyte, which compose in the inner dermis of our skin.
So now we know that the skin have all those three cells. But how can we obtain the stem cell? We actually can obtain it by using any somatic cell, any actively mitotic cell in our body. Since we are going to make a new skin in this context, so we might as well use our skin cells. All these three cells, when exposed to a transcription factors, OCT3 or 4, KLF4, and the others, it will cause a mutation on their gene. Those mutations will lead all these cells to revert back To what? To their original stem cell state. In this state, they can be made into any other type of cell. But as you know, we're making skin here, so we're going to have to revert to make this into those cells. How? How can we do it? How can we specifically make fibroblasts? How can we specifically make the keratinocyte or the melanocyte? We cannot just implant it and, oh, I hope it becomes fibroblast. No, we cannot do that. How can we do it? My friend will tell more about it.
So, to make the specific cells from the stem cells, we will give the specific conditions and transcription factors to the stem cells. So, for example, to make the keratinocytes, we will give the low oxygen concentration in mitomycin C. To produce the melanocytes, we will give FGF28, WNT3A, and other transcription factors. To produce the fibrinoblasts, we will give the FGF2. So, with all of these cells, we can then transplant it to our injured tissue to potentially provide the original appearance we once had.
That was the example of the application from induced pluripotent stem cell, which can be used to create a new skin. Now, you do notice that induced pluripotent stem cell have been existed from back then since 2006, and it has been through many kinds of development and also researches. Now, the main interest of this year is about to find a cure of neurological disorder, to control and prevent unwanted effect from the usage of the stem cell itself, and still the very same topic from back then, to create a new organ donors.
Thanks for watching our video. Don't forget to like, comment, and subscribe.
Melanocyte. Kok gue lupa nyebutin ini. Ini bisa tinggal aja. Tadi kok jadi gini sih?
Lim, gua bunuh Lim.
Reverse apa sih? Gue di reverse apa sih? Reverse KLF4 and others. Kayaknya salah sih. Iya. It must be given. Kenapa?
Stop.
Induce potential stem cells.
Stop.
Induce pluripotent stem cells.
Stop.
Outer layer of their. Kok jadi gitu ya? And these cells can promote